everyone. I hope you're coffeeed up. I'm really thrilled to be able to introduce our next keynote speaker, who is James Lendemer. James Lendemer is Associate Curator of the Institute of Systematic Botany at the New York Botanical Garden, and he's also a faculty member at CUNY Graduate Center. Since he will be in our subsequent panel, I will not introduce him farther because I'll give him the chance to do that later. James, are you ready?
Yep, I'm unmuted, I believe. Should I share my screen?
All
right, the dreaded moment. Great, you can all hear me and you can all see this, correct? That's thumbs up. Great. Should I go ahead and start? Yep, okay, great. Thanks. So again, thank you so much for having me today. This is really the exact kind of panel and workshop discussion and audience that I really enjoy speaking to, which is mainly the people that want to learn more about lichens and use them for various purposes. So I suspect that this talk, and let me know incidentally if my internet goes out or if you can't hear me or something like that. So I suspect that this talk might be a little bit of a departure from sort of the others in this workshop. I know it has, it will be because I've been listening to them. And it's because it's more about sort of life on Earth and the organisms and their potential applications rather than current applications or sort of attempted uses in biomaterials and fabrication. I still think it's important though, and that it'll be helpful because the fungi that I'm going to talk about, which are lichens, are filamentous fungi that have very different physical properties and biological functionality compared to those that are often used in biomaterials. And it's really clear that one of the issues with industrial applications of fungi, especially in biomaterials and fabrication, stems from issues that are associated with interactions, wanted or unwanted, with microorganisms and other fungi. And, you know, I think lichens could really provide raw materials to address these problems, both conceptually as sort of sources of inspiration, if you will, and also physically through molecular biology and physical properties, physical structures. So before I start, I'll just let you know that at the end of the talk, I'm going to drop a couple of papers into the chat. One is a recent publication from my colleague, Jessica Allen and myself, that calls for sort of a broad reconceptualization of these organisms. It was just published in Trends in Ecology and Evolution, and the link actually lets you directly access the article, so you don't have to deal with the paywall. And then the other is a recent review of a lot of the aspects of the lichen symbiosis and sort of recent advances in that. And that was published a few weeks ago in the New Phytologist. So two useful resources that I think would be really helpful for people that are interested in sort of trying to move forward with thinking about how to adapt things from these organisms to the systems that they're working in. Great, so with that, I will move forward. So first, a little bit about sort of where I'm coming from. So I've always been in love with the natural world. I hope many people here are as well. You know, even as a kid, I was really curious about plants and animals around me, whether it was in inner city Philadelphia, where I lived and grew up, or in rural Maryland, where my mother's family lived. It really seems to me that regardless of who you are and where you are, what your background is, what your community is, it's impossible not to wonder why the world is organized the way it is. And that's what really drives a lot of my work and my research. So what forces underlie the patterns that we see in nature? What accounts for the species that grow in a place, in a forest, or the diversity and abundance of epiphytes like ferns and mosses and lichens on a single tree. Our perspective of life on Earth, nature, and ourselves has changed radically over time. And increasingly, we understand that living things are really interconnected with each other and their environments in unfathomable ways and complexities. And it's understanding these processes and this interconnectedness that's key to successfully developing biomaterials and other industrial applications, especially for microorganisms at scale. So I think it's really important to take a huge step back from all of what we've been talking about today and think more deeply about the organisms that we're focused on, in this case, fungi, and how the things that we aim to utilize for new applications have exactly come to be. And it's really important to just step back and think about the fact that countless events and interactions have assembled the world around us, right? Life on Earth, Earth itself, and even the whole universe has really been shaped by patterns and processes that play out over scales that are largely imperceptible to us. Change has always been constant, and global ecosystems have been dynamic. Yet the remarkable forms and frameworks that we see in the natural world, at least as we perceive them, they reflect this really long game of geologic time. And I think it's really hard for us to conceptualize this because it means that what appears to be immutable and static is really the opposite of that. For instance, this long view requires us to understand that 20,000 years ago, the Earth was a much colder place. Glaciers pushed well south of their current bounds, which disrupted tremendously communities of plants and animals, forcing them to move and adapt. And as the glaciers retreated north and the climate changed again, species moved, species of more northern areas had to migrate up the mountains in the Appalachians to survive at latitudes well south of where they otherwise could today. So for instance, that's why if you look at a mountaintop boulder field in Pennsylvania or in Virginia, we see that in the shelter protected nooks and crannies, there are species that really occur much further to the north in the boreal forests or in the Arctic. Our perceived present has been assembled over these really vast timescales, and how exactly life has come to be adapted and organized from broad cross-cutting trends to individual specific processes of molecular development and evolution. That's really central to harnessing the full potential of these organisms in applied biology and industry. Now, to me, the central theme of life on Earth is really something that always comes back to this idea of change. And lives and ecosystems all around us reflect the coalescence of time. So mountain forests of Appalachia, they developed over hundreds of millions of years. Palm hammocks on the Atlantic coast, those have tropical species that are at the northern extent of their ranges, and they reflect a much warmer past. So there's these beautiful sluggish swamps and bays along the Atlantic coast that really developed to withstand and monopolize dramatic changes in water level and flow. So change has always been constant, and the pace and scale of change has increased rapidly with the development of what I think we would conceptualize as modern society. Its technologies, its attitudes. We've precipitated and created dramatic changes, disrupting, altering, and really even creating relationships between individual organisms, populations, species, and communities. So introduced pathogens decimate keystone species, air pollution and acid rain damage organisms and change the ecosystem chemistry. Resource extraction, such as logging, has extirpated species, while new and unusual fire regimes create challenges even for species that are adapted for burning. Extensive monocultures have replaced productive biodiverse ecosystems, and they're supported by altered hydrology and chemical applications. Wetlands are ditched and drained, and rising seas inundate, and saltwater intrudes into fresh and brackish areas. Severe storms erode our coastlines. The world is undergoing this great dramatic reorganization being driven directly and indirectly by us. Now, that isn't to say that there aren't many extreme and rapid events in Earth's history, volcanic eruptions, massive wildfire fires, flooding, and impacts with extraterrestrial objects. Those sorts of things have happened, but it's clear that the change that's happening now is really happening at an unprecedented scale and with an unprecedented speed in ways that are really perceptible to us within a single lifetime. And this has huge implications for individual organisms, entire species, and ecosystems. And those can span the whole gradient from sort of what we think of as nature to completely novel human-created environments. And I'm sure everyone has the understanding that life on Earth is remarkably diverse. There's hundreds of thousands of plant species. There's millions of animal and fungal species. and that only begins to touch on this like fathomless universe of microbes and viruses that exist. So each species is made up of a population with varying numbers of individuals. And historically, we thought of and studied those species and individuals largely in isolation. But now we know that everything does not exist in isolation, and that interactions between and among organisms, as well as their environments and other non-living things, are what drive everything in the world around us. Moreover, the occurrence, diversity, and abundance of organisms is really not driven by any single factor, but instead by vast suites of them that are intermingled with time. So it's, not just predators and prey or plants and pollinators, but it's everything from the microbiome inside something to the climate and the soil. And this all makes a lot of sense, I think. But this profound impact that this shift has had on science and society is really difficult to imagine. To me, so many of the issues and challenges that are associated with using fungi in biomaterials and more generally for biodesign and biologically informed sustainability stem from our nascent understanding of the central role of interactions, especially between organisms, and how they play out in patterns and processes that ultimately result in the physical or chemical properties that we kind of want to harness, adapt, or emulate. Today we're really focused on fungi, but I think it would help everyone to have a bit of background on the natural history and history of science of these organisms, because it gives a bit of perspective onto some of the challenges that you've been discussing today. So fungi are cryptogams, and this might be a new word for a lot of people here, but the root of it comes from hidden sex or hidden gamete. And this is a term that's used to refer to all of the botanical life forms that reproduce with spores rather than with flowers or seeds. So this includes all sorts of things: bacteria, plant-like organisms, fungi, which are evolutionarily separate from plants and animals, and all sorts of other kinds of microbes. Now, this might seem totally tangential, like after everything you've heard today, this might seem completely tangential to you, but these things are really all grouped together despite how dissimilar they are to each other, because early scientists actually couldn't understand them very well. And this legacy really stems from the lack of understanding or appreciation of non-typical and non-traditional anthropomorphized reproductive biology. And it's why knowledge about these organisms and hence their implementation in applied fields is really limited in many ways compared to various other types of plants and animals. So the institution where I work, the New York Botanical Garden, is somewhat unusual in that it has a legacy of inclusivity and in many ways equity for these kinds of organisms like fungi. So we have over a century of nearly unbroken study of these organisms, and in many ways that's due to the pioneering vision of Elizabeth Britton, pictured here, who was one of the founders of my organization, who was herself a specialist in mosses. It's this century plus of sort of inclusive and broad ranging study that's allowed us to assemble one of the major primary resources for understanding these different groups of organisms, especially fungi. And our work and the amazing resources that we've assembled over this long period of time are really central to a mission of training, education, and interfacing with all sorts of communities and stakeholders. So many people think of natural history museums and people like me who work at them as sort of stuffy academicians who work in sort of isolated, dusty old rooms. But the reality is that we have so much of the foundational experience and data and knowledge that's directly relevant to unlocking the potential applications for fungi and other organisms. And so that's why I'm really excited to speak with you all today. So biotic interactions are interactions between and among organisms, and these interactions drive a lot of the world around us. Fungi are among the most biologically diverse organisms on Earth. There are millions of species. We've heard that already today. They're literally everywhere in both diversity and abundance. They're on you, and they're in the room, and they're on every surface in the room. They are abundant and diverse in deserts, in rainforests, in old growth forests, in cities, on land, and in the sea. They are everywhere. They also interact with everything. They are the dynamos that are essential for ecosystem functions. And much of that, it's all made up of these tiny hyphal threads that they use to spin these shapes that are truly remarkable, varied, and beautiful. And these hyphae knit together the natural world, and they transfer nutrients between plants. Some fungi foster growth and creation, others hunt and destroy animals, still others decompose and recycle. The point is that they really span all of the possibilities of life. They're pathogens, they're parasites, they're commensals, they're symbionts. They do all of these different things. So over the last decade, fungi have really emerged into the zeitgeist, in part because of the visionary and transformational work of a relatively small group of people who recognize their potential. And so mycorrhizae are fungi that form intimate relationships with plants, and they're enjoying a particular moment of really extreme attention. About 80% of plants on Earth associate with these fungi, forming mutualistic symbioses on, in, and around their roots. The fungi obtain the carbohydrates they need, while the plants get the water, minerals, and nutrients that they need. The mycorrhizae weave together the natural world and the unnatural world in some ways, and they really form this imperceptible virtual network of everything, connecting plants of the same species and different species, shuttling nutrients, signaling data like fiber optic cables. To me, this is amazing, and we had almost no idea that this was happening, 10 to 20 years ago? Much of the vegetation on Earth wouldn't exist without these fungi, and neither would most of our most important agricultural crops. So lichens are similar to mycorrhizae in that they're another major group of mutualistic fungi, but rather than connecting plants in this sort of botanical internet, lichens associate with algae and cyanobacteria for the purpose of obtaining nutrition. There's over 20,000 species of lichens, and their unique lifestyle, which involves an internal food source, has allowed them to advance into nearly every available niche on land. So they live in the driest deserts and the highest mountains. They form the dominant vegetation in parts of the Arctic. They were even exposed to the vacuum of space and somehow survived unscathed. And as my colleague, Dr. Jessica Allen, and I advanced in this recent paper in Trends of Ecology and Evolution, we really believe that lichens have the potential to serve a wealth of applications in science and industry, and that we're really at this sort of pivotal moment where it's finally possible to achieve that, for them to sort of become the next mycorrhizae or the next sort of ideal fungal system for study and for application. The incredible thing about lichens is that they are persistent, stable associations with consistent novel morphologies that arise from obligate symbioses. So if the fungus could grow on its own, it would look like your everyday mycelium. If and when the other partners grow on their own, they would look like smears of green or gray or brown on a rock or a tree. But yet, when they get together to form lichens, which at least the fungi have to do to survive, they produce completely different, highly organized, complex morphologies, which is just remarkable. And these structures consist primarily of layers of fungal tissue with intermingled bacteria and microbes, and then also a single layer where the photosynthesizing partners are intermixed with the fungus and other associated members of the microbiome. So these fungal systems, these fungal symbioses form three main types of morphologies that are similar to the growth forms that plants can take. And this is just helpful for thinking about their structures and their overall macromorphology. So the first are the most simplified. Those are the crustose lichens, which are essentially bilayered, and they completely adhere to their substrate that they grow on. They're really good binders of things. The second are the leaf-like lichens, called foliose lichens. They're structured more like a sandwich, and they're anchored to their substrates with hook-like attachment organs. And then the third kind are the fruticose lichens, which are more shrubby and three-dimensional. And some of these are particularly noteworthy because their morphologies allow for significantly increased surface area for absorption. And also their branches, whether they're erect or drooping, have special properties that increase things like tensile strength. So they really have they've solved a lot of problems in nature and created really remarkable structures that are incredibly persistent and robust. I heard earlier people talking about how, mycelium on its own is kind of like trash substrate, trash material, but lichens are in many ways the opposite of that. They've solved that problem. Lichens are not trash. So when we look at a lichen, what we're mostly seeing is the expression and the structure of the dominant fungus. And every time we see a lichen that looks the same, it's the same fungus forming the association. So that's why when we talk about lichens, we refer to them by the name of the fungus. Now, traditional views of symbioses in nature were based on this really simplified binary one-to-one association between species. So when we traditionally or historically thought of symbioses, we thought it was one thing interacting with another thing, very binary, very simplified. So in this case, this Anaptychia palmulata fungus was maybe always associating with this algal species Trabuxia glabra. But now we know that the situation is actually much more complex and dynamic than that. So for instance, when the species grows in New York, maybe it associates with this alga, but then maybe when it grows in Virginia, where the summers are hotter and winters are a little less harsh, it might associate with another alga. And maybe in South Carolina, it associates with a different alga altogether. So one species can associate with different photosynthesizing partners, right? Like potentially this one species might associate with these three. This is hypothetical. The numbers are much larger and also much more variable. But you'd expect that in biology. And so that's one possibility. But the reality is that even now we're seeing it's even more complex than that, and that what we perceive as a single individual can host multiple of these partners. And again, I think that's really logical, but it's really only recently becoming clear just how common and widespread this phenomenon really is. So, the question I think a lot of people pose, and what, this is sort of, it's in some ways a basic question, and in some ways it's a sort of philosophical deep question. What is a lichen? And this modified version of a figure from that Jesse Allen made to explain this, I think, sort of gets at it. A lichen is a really dynamic system of living and non-living things that are organized within and around the scaffold created by a single dominant fungal species. And that it also includes at least one, if not more, photosynthesizing partners. And then it also includes a microbiome of endoglycinic bacteria, and then also endoglycinic fungi beyond just that main dominant fungus. And importantly, this microbiome of bacteria and other fungi includes organisms that run the gamut from mutualists to commensalists and even parasites and sometimes pathogens. In addition to these things, then there's also all these microscopic animals that live in and around the lichens. And then there's a huge array of metabolites that are involved in signaling and numerous other important functions. So what is a lichen? A lichen is this really complex and dynamic thing. It's a hub of interactions between all sorts of organisms and the environment. But it's even more complex than that. So we perceive lichens as splotches on trees or rocks, static, unchanging, benign, and slow growing. But they're not at all that. They're not at all what they seem. And in fact, what appears to be a single individual can instead contain one or more species and genotypes of photosynthesizing partners, together with a really diverse array of microbes. And while there is only the one primary dominant fungal species that forms each given lichen, there can be multiple genetically distinct individuals imperceptibly intermingled inside what looks like a single one. So, this remarkable dynamic complexity provides exponential dimensionality and explains how such seemingly small, sort of static things have proved so adept at adapting to change over geological time. And a variety of partners and with differing specializations and environmental suitabilities. You can imagine how that's quite helpful in a changing world, and incorporating the sort of spectral multi-membered lifestyle into living biomaterials, especially ones that are living, could render them more robust. So then what is an individual when your perception of an individual can contain many or one of your own kind? What is a population when interbreeding individuals exist intertwined together, but spatially separated from each other on the landscape? And if you can't move, how do you find new mates and how do you find new partners? Lichens just defy and challenge many of the traditional classifications and frameworks that we have in biology. And then you can extrapolate that out for what that means for a whole community of species. So different species of lichen fungi might associate with the same alga or with different algae and different microbes. Different lichen species growing in close proximity might have more similar microbiomes and partners than two individuals of the same species that are growing miles away from each other. It's a paradox. It's a paradox of flexible specificity. Species and individuals seem flexible, right? Able to associate with suites of relatively closely related partners, but at the same time, they're highly adapted to where they live. And so this remarkable strategy means that, diverse lichen communities with diverse pools of partners and microbiomes, they reinforce each other and they foster even greater diversity and abundance. But at the same time, they're highly interdependent and highly interconnected. So suites of lichen species form guilds of shared partners, and they rely on each other for their collective survival. But they're also impacted exponentially by the loss of one species or even one individual. So here I've identified a small selection of species that are growing on a tree branch in North Carolina. Some of these are likely sharing cyanobacterial partners and other bacteria, but their remarkable capacity to function as natural sponges, absorbing and releasing moisture back into the environment, means that they are also able to foster the environmental conditions that reinforce the growth of even more lichens. We like to say lichens beget lichens. So lichens essentially swamp the environment with their clones in order to reach a critical threshold that they need to have an available pool of suitable partners and also to have stable and suitable environmental conditions. And so a way to imagine this is if you take this species, Candelaria concolor, commonly called the candle flame lichen. It's the most common yellow one you see on trees in cities and likely around you in State College. Like many species, it has evolved specialized mechanisms that allow it to bud and fragment. This is a sort of controlled deconstruction. That's one way to think of it, wherein the lichen exposes itself and forms a fragment, little cotton ball-like propagules that include the fungus and all its partners and associated microbes. These are dispersed into the environment. They fall down the tree. They blow short distances in the wind, attached to the fur of a passing animal. They're even dispersed or splashed by a raindrop. And when it lands in a suitable place, it becomes lodged and it establishes a new lichen that's presumably identical to its parent. Imagine now that this process unfolds almost invisibly to us, too small to easily see, with growth occurring over years and decades. But also this is happening at scale, with a single lichen constantly shedding hundreds or thousands of potential future lichens into its surroundings every day. This is how they swamp the environment and establish a ready supply of partners for the right moment when they have sex and they're forced to resynthesize themselves from scratch. So this is, to me, totally amazing. But also then what happens when those pieces, instead of dispersing on that same tree, when they go a slightly further distance and allow the lichen to colonize a new tree, growing another identical community there. When that original tree blows down, falls down, dies, and its lichen community isn't eliminated, does the lichen die if a clone that's established on a different tree has survived? Under natural conditions, allowing for these kinds of processes to unfold sort of without their contraction and disturbance by human timescales, these fungi really have non-cyclical and non-linear lives. So they're a mosaic of species that you see growing on a rock or a tree, they could reflect thousands or hundreds of thousands of years of stochastic events. And this is why lichens are so diverse and abundant in environments and healthy environments, and why it takes so long for them to recover from disturbance and extirpation if they ever do. We actually have no observed case where we've documented full recovery of a disturbed or extirpated lichen biota. So when present at scale, in the environment, they naturally would be abundant, and they are essential to most ecosystems. So every tree and rock and square inch of soil should be covered in a blanket of lichens. And in abundance, they regulate humidity and temperature because they have these sponge-like properties. Hard to imagine with the scale of a single lichen, easier to imagine the scale of metric tons of them in the environment. So they're able to become almost completely inactive when dry, and then they persist in that state for long periods of time. They're only physiologically active when wet, and the naturally punctuated activity associated with normal moisture availability in the environment. Most environments are not equally moist and moist all the time. Instead, it's very cyclical and periodic. That sort of punctuated lifestyle or activity coupled with their rampant clonal reproduction means that they even have essentially indefinite lifespans. So they're abundant, they're diverse, or they should be, and they also provide really essential food and forage for just about everything from small insects to caribou. Birds use them for nesting materials. Everything uses them in some way. They even play critical roles in nutrient cycling and nitrogen cycling. And if mycorrhizae are sort of the invisible networks that link everything together, then lichens are the hubs of action in the world around us, and they can actually be readily seen and appreciated for that. So, they also, it's probably not surprising, have a huge, long history of use by humans. And so they've been integrated into belief systems all over the world on every continent. They've been used and are used actively in food, often in times of starvation, although not always. Some cultures have developed cuisines around them. They've also been used and integrated into many cultural practices, for instance, mummification in ancient Egypt. And then many of their uses actually relate directly to their chemical properties. So for instance, they were used historically in medicine for the doctrine of signatures. It used the treated lung ailments with this species lungwort because this species happens to look like a lung. They're used in modern medicine as well because many of their substances have bioactive, antifungal, and antimicrobial properties. And then also many of those same substances also function as natural poisons. So they've been used, the substances have been extracted and used as natural poisons in the past, although I would argue we have probably far more effective and easier to obtain synthetic poisons now. Now, most uses, as I said, most uses of lichens, past and present, derive from their diversity and abundance of metabolites. And one of the best examples of this comes from their use in natural dyes. So here you can see a time series of the beginning of extraction and production of a natural dye from a lichen sample. I did this last summer. And it might seem like this is possible to use lichens in this way and in many other ways for like a single person, but industrial applications and scaling up are really complicated by the fact that everything is harvested directly from nature. And that's, due to the growth and dynamics of natural lichen communities, it's basically impossible to imagine a scenario where you can actually extract these things from nature for industrial applications at scale and sustainably. It's not possible. And yet, if you go to, for instance, Etsy, you can see them for sale in quantity. And the classic example of how this is really not sustainable is that the lichen dyes were used historically in Scotland because they were one of the few sources of purple and red in nature. And so very quickly, local sources in Scotland were actually over-harvested. They had to start importing lichens internationally from Scandinavia. And very quickly, it became economically unfeasible and unsustainable, not to mention the fact that they had completely extirpated the supply of local lichens that were presumably performing essential ecosystem functions. Now, this might seem a bit far removed from the direct topic of today's workshop about biomaterials and biofabrication, but I think it's important to recognize that we see lichens used in all sorts of ways. Right now, mainly as natural products or for their natural products and natural chemicals, and also in arts and crafts and things like that. And even though the current uses might seem like small scale, the amounts of lichens that are being removed from the environment is actually quite high. And even though the many sources might claim that this harvest is being done sustainably, there's little evidence that that's actually the case. And so the reason that these things are removed from the natural environment is that we don't actually know how to propagate them on a large industrial scale. But yet there's this huge demand precisely for them, for their properties, for their chemicals, for things, and for just in general for their themselves, for the lichens, for bioremediation, for ecological restoration, for biodesign, and for architecture. So there's all of these diverse groups that want to use them for all sorts of applications. And I would argue why we really haven't figured out how to grow them and use them for these applications and really harness their potential is that there's been a lack of focused attention and funding, especially with interdisciplinary collaboration. So most people that study lichens are lichenologists, and then there's a disconnect between the lichenologists and everybody else who wants to study them and actually use them for things. And then second, I would argue that one of the key aspects to unlocking the industrial applications of lichens stems from aspects of their fundamental interaction-based biology. And we're only really starting to become knowledgeable about that. Like that's only becoming accessible thanks to recent advances in molecular biology and sort of more systems-based thinking. So, our present inability to readily manipulate and grow these organisms at scale in laboratory conditions reflects the more general paradox about lichens. On one hand, they are the best and most adaptable organisms on the planet, yet on the other hand, they're generally poor at adapting to the kinds of rapid changes that characterize our current period. And that's why lichens have declined substantially in diversity and abundance across natural systems, like here in this mature forest on Long Island, and in urban areas like here in Central Park in New York City. Now, while this current period of rapid disturbance and change may greatly impact many lichens, it does seem clear that some species will adapt and survive. And we can see that in New York City, which has like one of the best studied and longest studied lichen communities outside of Europe. And we can see, here's a graph of the number of species that have been found at various time points. It's amazing to have such good data going back to the 1820s. That's really unheard of in the New World. And here you can see that there's a clear decline in the number of species from when the city was smaller in pre-industrialization through its rapid build out, and then extreme period of air pollution and industrialization. But that this has been followed by a subsequent increase and return in species that are tolerant and adapted to novel environments and the skewed climates of urban areas. So it seems clear that what's happened here and arguably everywhere else is that, there was an initial build out that extirpated most of the species when there was large scale rapid disturbance, and then there was this period of air pollution that extirpated almost everything else except the most resilient species. But now with cleaner air and more stability, it's not like in New York City, we're tearing down everything and building a new Manhattan every day, right? Things are much more stable than they used to be, even though things are still being built, things are still being deconstructed, there is still change. It's very different and on a different scale than it used to be. Now that it's a little bit cleaner and it's a little bit more stable, things are able to return and are actively returning. And we have a pretty good sense that the natural processes of selection are what have to play out to allow this kind of return to happen. So for instance, this is a pollution sensitive species that was extirpated from New York City, Usnea mutabilis. This is it in all its glory in Maryland. One individual of this species was found in the Bronx in 2018. And another one was found in Brooklyn in 2020. And both of them were so small that we couldn't identify them to species with certainty without sequencing their DNA. But they had returned, and they returned to relatively large, naturally stable environments into cemeteries. So at the same time, we also tried to transplant this species back to New York City. And you can see this is what the transplants looked like in 2015. Within a few months, they did not look so great, and it was only a matter of time before they were completely dead. And this is a general trend that's been observed in translocation attempts to urban areas and built environments numerous times in many different places. And so really what all of this gets me to is what I think is at the heart of this workshop, which is that we're living in this unprecedented time where life is really being forced to adapt to new and unusual challenges. And we're also trying to use life on Earth to aid in our adaptation and mitigation strategies to continue to survive ourselves. And key to succeeding in this is really untangling and harnessing the strategies that other life forms, such as lichens, are themselves using to adapt to these new and novel conditions. So on the left is the Palmerton Superfund site, where pollution from zinc smelters killed most of the vegetation within miles of Lehigh Gap in Pennsylvania. Some of you may have been there or be familiar with this area. There were no lichens there in the 1970s, but by the early 2000s, a number of lichen species had actually managed to adapt and recolonize the area on their own. And so that's no different from having a community of lichens that have somehow established and thrived on a storefront awning, on 14th Street in Manhattan. The whole point of what I hope I've sort of inspired everyone with today, and what I would hope we can talk more about, is that we have this opportunity to harness sort of the Anthropocene resilience of other organisms. Specifically, I would argue lichens, because of their sort of multi-organismal and stable symbioses and really stable physical properties. We can harness these things because that's going to be essential. We need to do that because it's going to be essential to our own resiliency moving forward. So with that, I will just thank some of the many collaborators and people who have made my work possible and this work possible. And I appreciate, again, being invited to speak to you all today about something that was very different from everything else you heard. And I guess there's a few minutes for questions, maybe. I'm not quite sure.
Thank you, James. That was wonderful. And just as an aside, you win the prize for the most precisely 45-minute talk so far. Well done.
What was it actually 45 minutes? Wow.
I think so. Yeah. Right. So I hope you enjoyed this freewheeling presentation that James gave, and I am hoping that it inspires you to ask similarly freewheeling kinds of questions. We're here on the Wild West in terms of fungal biomaterials. There were, fungi are enough of a black box, but lichens are, we can't even really grow them yet, but we are starting to be able to understand their properties and their characteristics in a deeper way. So do we have any questions on the chat?
And I will say, whoever figures out how to unlock the potential of these things stands to make a huge transformational contribution.
I agree. I hope it's going to be you.
Me too.
James, I think
it'll be me with others. Yeah.
I think you can unshare at this point.
Oh, sure. Sorry. Yeah. I have a tendency to overshare.
Yes. Okay, great question.
Oh, I'll drop those. I'll also drop those links into the chat.
Terrific. Hi, thank you so much. That was such a great talk. My name is Sharifa Crandall. And, I was wondering, when you showed towards the end, that picture of the Anthropocene and the Superfund site and then the awning, and I was wondering, have you or has anybody else sort of looked at, what makes the lichen that colonized those spaces different, from the ones that are, say, just in a forest, for example, where there's plenty of resources and there aren't weird chemicals and, things like that.
So that's a great question. I also feel like everyone always says, that's a great question, but that is a fundamental question that is, so on one hand, many species could probably recolonize these areas. It depends obviously on what happened to a place, but lichens are naturally pioneers and early colonizers. The thing is that there's just a delay in their colonization because they have to, to anthropomorphize this a bit, they have to figure out the partnership that's going to help them survive under those conditions. So presumably it's a matter of species that are already adapted to rapid colonization through clonal reproduction, which more than half of them do. And then in addition to that, it's like you've got that going on, but then you have to have the right mix of microbes and partners that will allow you to survive under whatever those changed conditions are. So in this case, it happens to be, super high amounts of zinc, cadmium, and lead in the environment. But also lichens are fungi, so they're very well adapted to sequestering all of these kinds of things and chelating them inside of their bodies. That's why they could in many ways be probably used for bioremediation. So, that's sort of, and at a more fundamental, so that's the sort of macro scale picture of that, but at a more fundamental level, we think that there have to be sort of molecular genomic adaptations that are happening that are, allowing certain combinations of things or certain individuals to be able to survive in some of these places and to be able to return. And it's just all they have to do is something has to adapt to that condition or be able to survive in that condition. And then that clonal reproduction will just allow them to swamp the environment. And that's what we saw at Palmerton. All the species that recolonized the area, I think almost all of them were clonally asexually reproducing things. And at least one of them was something that's actually known to be highly tolerant of zinc. So hope that answers the question.
All right, thanks. Sharifa, did you have a second question? Okay, great.
It's the same answer for recolonizing urban areas. Everything was extirpated, and then there's a lag in the period for when until something comes back. And when something is able to come back, there's another lag because it has to swamp the environment with enough propagules to have enough of the symbionts around for them to be able to reestablish.
Thank you. Is there a chat question?
Yep.
Okay. Can you?
Yes.
Okay. So I'll read out loud. Hi, James. Thanks for the presentation. I knew so little on lichens before. You mentioned in their first form of crystose, they are good binders. Can this be applied like mycelium, or do they only grow on living photosynthesizing matter?
Well, they're not. So the lichens are only using the things that they grow on essentially as a substrate to be to physically latch onto. They are having some kind of connection with the things that they're on, the rock, or they're anchoring into it some way, but they're not actually interacting directly with their trees if they're growing on those. So they can live on whatever surface it might be. They'll grow on plastic, on metal, they'll grow on anything that they can possibly grow on. It doesn't really matter. And they can bind things together. It's more a matter of they can't be sort of like integrated into a material itself because they have to be exposed to the light. But they can grow relatively rapidly. I mean, whatever rapidly means in this context. But yeah, no, they can certainly bind things together, but it's more of a slow process in nature. So again, this is one of those things where like they have all of these properties. The question is, what do you do to either speed that up or scale it up or do something, use it in a way that gets you to the point that you want. But yeah, they don't have to grow on things that are living because in fact, they don't actually grow mostly on living things. They're growing on the outer surfaces of living things that are actually like dead. So, so when it's on the tree, they're growing on the sort of outer cells of the bark and stuff that aren't really alive from the tree standpoint.
Oh, great. We've got another question back here.
Hi, my name's Maya. So this is going to be kind of a weird question, but
no such thing.
I was just kind of curious. So you were talking about like lichens as hubs of action, and then you also had previously mentioned like, mycorrhizal interactions of plants with fungi, and then I know there's also like bacteria underground. So I was wondering, are there like, because you were saying, plants, when lichens grow on plants, they're not really interacting, but there are fungi that are interacting with plants. So like underground, are there also like plant fungi bacteria hybrids that are hubs of action similarly?
I mean, I guess you would think of them, you could think of them similarly. It's just that the lichens have that all happening in them. right? Like they're actually forming the structure within which that's happening. Whereas I guess with mycorrhizal fungi, I tend to think of that as like the soil is the substrate and the mycorrhizae are just sort of interlaced all throughout there in various amounts of abundance. So it's a little bit different because the lichens are really the thing that has everything happening in and around it, to me.
Okay, one more follow-up question to that.
Go
Are
for it.
there like endosymbiont bacteria? Are there also endosymbiont fungi at all with plants?
You mean with plants or with the?
With plants.
No,
Like-
yeah. There's endosymbiotic fungi and everything. Fungi are everywhere.
Okay, thank you.
All right, thank
Yeah,
you.
there's a great field of like endophytic fungi. There's actually there's some really cool work that's been done and it's actively being done by Betsy Arnold's lab and Jana Uren's lab and Francois Luzzoni at Duke, Arizona, University of Arizona in Tucson. That's looking at endophytes in plants and sort of how they are actually more structured and spaced in the environment, and how actually there's these sort of networks of them that also are in the lichens. So the same things that are in the plants are sometimes also in the lichens, then also in like soil and liverworts. It's all much more connected and much more involved and dynamic than anyone anticipated before. But I think that's what we would think of if, that is nature. And like, if we can appreciate and really understand that, that's going to help us in translating that to these kinds of applied fields like biomaterials, right? Like you're trying to recreate that essentially, right?
Great. And just spoiler alert for Maya and James too. We have a local endophyte expert here. That's Sharifa Crandall, and she's going to be on the panel in just a few more minutes. Do we have? Okay, great.
So I also didn't know much about lichens before your presentation. Thank you. So this question also may not be a good question. I don't know. But
Every question is good.
Okay, so we saw in the previous talks and also from my personal experience in my lab with my grad students that contamination is a big issue in making fungal biomaterials. And it can be like when there's different types of contaminants, when they grow, it's difficult to obtain the material itself because there's a competition going on. And I wonder if lichens could come into play and, help maybe like eliminate the contamination by creating an ecosystem there? Or are there types of lichens that can serve for this kind of purpose? Again, I don't know if that's possible or I just wanted
No, that's
to
a great, that's like an exact logical extension. So the thing is that often when people are interested in sort of thinking about applying lichens for a purpose, it's often in this sort of literal sense of like, can we take a lichen and do a thing with it? And what I really encourage people to do is to step a little bit back and think, well, okay, it's more like, can you try and replicate the function or use something that the lichen has for the purpose that you want? And so the whole problem is that you have all of these mycelia that are growing sort of essentially just out there in the environment that are doing things, and you're trying to grow them in a very different condition where they're sort of in isolation and you have all of these contaminants that potentially get into them. Lichens are like the fungi that have built their own sort of sterile laboratory, right? Like they have devised a way to keep all of these other fungi and microbes that will contaminate them and kill them out. And that's all of these secondary compounds that they have that have antimicrobial and antifungal properties. And we are in the process of starting to understand a lot of those biosynthetic pathways. And the real goal is to get those substances to be able to be produced in yeast systems or something like that. I don't think that that's really come to fruition, certainly not like at a scale yet, but we're starting to get there. And that's potentially what you're talking about, but also has a ton of additional applications because all the pharmaceutical industry would love to get a hold of many of these compounds. It's just that you can't get enough of them to do anything with them in nature. So the question is, how do you synthesize them?
Great question, Benet. Thank you, James. And we're going to actually return to this question of interactions between biological components of a living system in the panel. But before we do that, I have one more question. Is there anyone else in the audience with a question? Okay, I'm going to ask a very nerdy microbiologist kind of question now. Can you talk a little bit about, so cyanobacteria are from the bacterial domain. They're single-celled prokaryotes. That's quite a different animal, so to speak, than an alga, which is essentially, you know, a eukaryote with a nucleus and a former cyanobacterium. So it's like the cyanobacterium is inside the algal package, and then the alga is inside the lichen package, right?
No, it's separate. So in
Okay.
what the lichens do is they either associate with cyanobacteria as their partners, or they associate with green algae as their partners, one or more kinds. And then some species associate with both. But when they associate with both, the cyanobacteria are most often encapsulated in a nodule, and the rest of the lichen has the green alga in it. But then there's also some of them where just the same fungus can either associate with either the cyanobacterium or the green alga, or there are different stages in its life cycle. Exceptions to everything, but the general rule is it's one or the other, and most things associate with the green algae. But again, green algae are really diverse, and they have, you know, they have sort of different functional ecologies too. So, you know, not all lichens that associate with green algae are equivalent, and that's actually something that we're only really recently starting to come to terms with, right? Like, I mean, it's so funny because the lichens are sort of the nexus of all the understudied things about all the groups, right? Like, you know, the fungi themselves were understudied, but then it's like, well, you have to actually understand these microscopic unicellular green algae, which themselves are something that, you know, almost no one has studied in great detail. And then the ones that are in lichens, are they different from what's in the environment? And it's really, it's just,
James, you and I have to avoid, well, we have to talk in private because we could probably go off for a long time about this topic.
But it's amazing because we are at the point where we can understand these things. The technology has advanced to the point where it's possible.
We just need dollars and people.
Yes. Well, isn't that always the problem?
Well, no, because technology sometimes isn't there, as that, you know, that point has been made, I think, already today. So yes, it's an important moment. We have the technology, but not the money and the people right now.
Yep.
Okay, so this is a good moment to switch into panel mode. And Sharifa, would you come up to the front? And John, is John here in person? Okay. Is John on? Okay, I think we may have lost John for today. I'll try to stand in and Carolee, if you want to weigh in on things, please do. Oh, and Maria also. Yes. Right. Well, we have many experts in the room. So John Pecchia is the director of the Penn State Mushroom Research Center. It's a center that's very well funded, in part because of the huge edible mushroom industry in Pennsylvania. I think I learned, correct me if I'm wrong, anyone who knows, but I think I've learned that the biggest agricultural crop in Pennsylvania is hay, and that's because it's used as a, what is it, Carolee? Mushrooms are the biggest commercial crop in terms of money. Yes, and then what's the second biggest one? It's what mushrooms grow on. Yeah. Anyway, it's a big business, and so what we're missing when we don't have John in the room is this sort of commercial liaison and the economies of scale that you get with growing fruiting bodies, which, by the way, that's a very different exercise than growing mycelium for leather or biomaterials. So yes, you know what? Great idea. Sharifa's just said, let's have a little bio break. So let's get back here in like 5 minutes or so. Does that sound all right? Okay, great.